password.
Oh, okay.
Because I don't know, like,
Oh, no.
and my brain isn't like, you do have to switch.
Oh, really?
Or you
Yeah. Oh, well.
or someone.
No, he was quite
Okay,
okay. He's like,
about
oh, yeah. And then obviously, yeah. So after the point, okay. So with the bio, yeah.
Because this is really so I got that. People works in the Berkeley Research Development is the assistant team for UCSF. So I'm projecting it as soon
The liaison between the campuses.
and he has a budget.
Just asking how widely this is.
I think all the places it's really this small. This is really
It was widely.
Oh, yes. Just read out all the bio.
I need to check this exact distribution costs. But we went through chairs each other. And so he's really concerned about people. At that time, was actually well, right now he's covering Saturn. Every 3 years, he's going to pick up more less time. Yes. And then this is really to our director
I have collaborators.
So I'm going over to.
But I did not project this to students.
With boss Marshall should be done by 11. Maybe today. So,
School related.
All over the group yesterday and asked.
Wendy listed.
Some practice.
There's the common.
yeah, I'm still have the form.
So it's called UCSF.
Right, so we'll do lunch now. I think I don't think my flight's going.
Oh, yeah.
So, I'm actually San Diego, and then I'm going to Hawaii.
Yes.
Once a while.
Yeah, that'll be really fun.
Like that.
I bet, yeah. That's a really long.
So,
Yes. I was just in Hawaii for the very first time, and learned that not lots of people from the East Coast can fly the chemistry.
The time difference is killer.
Yeah, that's all.
Yeah, I mean, it might as well be international.
That's how it should go, but, okay, I don't know.
Kind of like trying a machine that makes.
Oh, okay. Yeah.
Know what that is called.
So you haven't been on
Oh,
Ribosome.
yes.
No,
Yeah, that's really okay.
I mean, there's just
Oh, yeah. I think it's.
Oh, I think she mentioned that now. So, yeah.
But I think they were.
Good.
I felt like I have to let my knee go. This bit here is just a cartoon way to show the approach.
Hi, welcome. Thank you all for being here this afternoon. My name is Paige Sorensen, and I manage the Team Science for Research Innovation program in the UCSF Research Development Office. So on behalf of the Research Development Office, I'm very proud to welcome Dr. Charlie Cunningham, who has graciously traveled from the National Science Foundation to be here with us today. Dr. Cunningham is a program director in the Division of Molecular and Cellular Biosciences. He has also recently taken over as the cluster lead for cellular dynamics and function. Since the previous lead, Dr. Greg War, has recently retired. Before his role at the NSF, Dr. Cunningham was an associate professor in the biology department at the University of New Mexico. Dr. Cunningham also just told me that he did his sabbatical here at UCSF, so he's a bit familiar with our university and campus. So without further ado, please welcome Dr. Charlie Cunningham.
Thanks, Paige. I have a tendency to walk while I talk, so if I'm over here and I'm speaking like this, I hope you can hear me. If not, they said I can be mic'd up, but hopefully we won't need that. So thanks again for inviting me to tell you something about NSF. Outline of the talk, what I'm going to do is I assume that there would be a lot of younger people here today who do not know much about NSF, probably more familiar with National Institutes of Health. And so I'm going to give you some overview of the NSF structure that might give you some idea about where you should be applying. It's not necessarily the Directorate of Biology where I'm from. There might be other opportunities there. I'm going to say something about the funding opportunities that are available, mostly for single PI grants or co-PIs. I'll say something about the NSF merit review criteria, again because it's very different from the merit review criteria that we would normally associate with NIH. And the process is different. And then just towards the end I'll use some time to tell you about what we look for, what makes a successful grant, but probably more importantly, you probably all know what makes a good grant, good ideas, but also what NSF causes you to fail and can cause you to fail. So how to write a successful proposal. So NSF against NIH. So there are similarities, of course, and one of the similarities that can cause a lot of problems is the overlapping scientific interests. So I'm from the Division of Molecular and Cell Biology. We fund cell biology. Clearly, NIH funds a lot more cell biology. How do I know who I should be going to? The answer is, in some ways, very simple, but if you think about it too hard, it can become quite difficult. The simple part is we do not fund medical science. We do not fund science motivated by medicine. And I'm only talking about bio at this point. I'm only talking really about MCBs, but that's generally true of all of bio. And so we fund basic research. So this basic science versus biomedicine in our missions. And so if you think of research as a continuum with the payoff for the patient at the end of the continuum, we're at the very start of the continuum doing the very basic research that might lead you to have enough preliminary data to write your first ROI. Okay? So we're at that stage before that. So as you'll find out, you should not be couching your research aims in medical motivation. And I'll come back to that. We differ in our review criteria, details of the review process. Again, role of program directors. This is again an important one. At NIH, the committees who read your proposals tell the program director, this is what we want you to fund in this order, and they have to go down that order until they run out of money. At NSF, the committees who review your proposals suggest to us what they would like to fund, and it's up to us whether to take that suggestion on board or ignore it. And so we have more or less authority to fund what we want. That makes a difference to you, because when you come to write your proposal, you have to realize that you're actually going to be writing that proposal for two audiences. You're going to be writing it for the reviewers in the room, but you also have to write it for the program directors as well. And that basically means looking at our solicitation and seeing what we want, because even the reviewers don't bother to do that. They just rank the things and, oh, that's great science, and maybe ignore the broader impacts, which we're going to come to. But they maybe just ignore the fact that we're looking for a bit more computational modeling than they're used to reviewing or what have you. So you're writing for two audiences. And then, of course, the big difference is the budget. We're $7.5 billion, NIH is $32 billion, notwithstanding whatever happens in the next budget, of course. So at a glance, $7.5 billion. And this is the number here that people like to latch onto. Approximately 24%, sorry, not that number, this number, 23% success rate. Yes, we have a 23% success rate, but of course the devil is in the details. We get a lot of stuff that we ask people to submit. Eager awards, for example, the hurdle is being asked to submit it, not the submission of it. Once you're asked to submit it, there's a high chance it's going to be funded. So that's the hurdle that you have to clear. Most people, though, are putting in proposals to panels, and I would say that in MCB, our funding rate is somewhere between 12 and 15% for investigator-initiated proposals, and that's probably the more important number, rather than this kind of false advertising at 23%. So 12 to 15%, and we are prepared to take a 10% cut in our budget. I don't think that number will fall too much if that happens. If it goes above that, things might get a bit more tricky. Down the bottom, I've got here half of program officers or rotators. This is just to let you know that, yeah, we're making these decisions on your grants, but we bring in fresh blood. About three of my colleagues are what we call rotators, where they come in for 2 years. And so they're bringing in fresh ideas, they're seeing fresh proposals often for the first time, they're telling us often what they would like to see funded, so we get a lot of input from the community. And this is the point at which I say, if you want, for those of you who are more senior in your career, and you think this is something that you might want to do, then we're always looking for rotators to come and work with us in DC, in Washington. So just a bit on the structure. We don't have institutes, we have directorates, and we have 9 of these. And the ones that are circled are the ones that you are probably going to be most interested in. Biological sciences, of course, but I'll also say something later about engineering and mathematical and physical sciences, because often if your proposal is more led by the chemistry, the physics, or the engineering, then with some biology attached, you want to be thinking about these agencies. The other nice thing is engineering, I know, and I think physics too, not sure about chemistry, but engineering definitely, is one of the parts of the agency that accepts medically motivated proposals. So they do a lot of tissue engineering, for example, being one of them, clearly medical motivation. So what I'm going to do is spend the next 10 minutes or so just drilling down a bit into that organisation. So we, Directorate of Biological Sciences, kind of cheesy mission statement, enable discoveries for understanding life, but again it speaks to basic biology. And we're divided into 4 divisions that are listed here, and I'm just going to say a little bit about the three of them over the next 3 slides, and then a bit more about molecular and cellular bioscience. So this one, biological infrastructure, exactly what it sounds like. Divided up into the part of the division that deals with research resources and the other part that deals with human resources. So a lot of the big budget stuff that comes out of bio comes from research infrastructure. So things like the national environmental studies, the NEON studies, which got a huge amount of bad publicity, cost about $85 million to start up, were funded out of there. And in some ways this was biology's response to, and a bit of big project envy. We were always looking at the physicists and the astronomers with their big telescopes and thought we want a bit of that, and so we set up NEON, and we got our fingers burned, and so now we're worried about doing that sort of thing again. But they also fund the ships, the polar ships. So they build ships, they fund research stations, they fund research collections, so museum collections as well. They also deal in human resources. So going just from the bottom, the research coordination networks, if you have a big idea, potentially transformative idea, and you need to build a network of your colleagues and your friends, then research coordination networks are probably the one that you want to be thinking about, because they're, what we're looking for is transformative ideas that also cross boundaries. So we would expecting you not to be hooking up with 4 or 5 of your colleagues in the same department, but bringing in engineers, bringing in mathematicians, chemists, physicists, as well as biologists, and building a network of individuals to tackle a single problem. The one that I really want to focus on is postdoctoral research fellowships, and the reason that I want to say something about that, and I've highlighted it in red to remind me to say that, is because this is where I think the NSF it has an area of weakness. And we can give you money for high school students, for high school teachers, for undergraduates, graduates, postgraduates, and for newly minty tenure-track professors and full professors. But the one place that we only address postdoctoral research fellowships, meaning that we give the money to the postdoc, not to the PI to employ you as a postdoc, but to you as the postdoc, are these postdoctoral research fellowships. And this is the only award that I'm aware of where you don't have to be an institutional PI to actually apply for an NSF award. If you're a postdoc, you can apply. Now, caveat, you have, depending on what you're applying for, you have to be 6 months or 1 year into your postdoc maximum. You really should be thinking about this as you're writing your PhD thesis and you've arranged to go somewhere else to do your postdoc. You should be in touch with your next supervisor and coordinating with him to apply for one of these research fellowships at that point. Having said that, the reason I don't think we do enough is that the program isn't far-reaching enough. There are 3 caveats to it. We will fund you if you're a minority PI, irrespective of the research that you're doing, if you're a minority. Now, they actually say, I looked at the call this morning, And they use two different descriptions. They say bringing people from underprivileged backgrounds into science, and then they also say minorities. Two different things, because if you're the first in your family to go to college and you're from a blue-collar background, then that, to me, would meet the underprivileged criteria as opposed to the minority criteria. So with all of these things, speak to a program officer if you're in any doubt about whether you're eligible. And the second part of this is if you're working with museum collections, you can also apply. And that part of it changes every few years and it's due to change again. So I would find out if they're thinking about changing that part and if they're going to change. But if you're working with biological museum collections, you can apply for this. And then the last part is the Plant Genome Initiative. If you're working and doing anything with plant genomes, I know that this is probably a predominantly UCSF audience, but I also understand that there were other schools present today. So if you're working in plant genomes, doing anything associated with plant genomes, apply. In fact, I would apply to the Plant Genome Initiative for as much as you can, because they're a well-funded part of the agency. They get relatively few proposals. So that's, and that applies not just to postdoctoral research fellows. So those are the three caveats about postdoctoral. And then I'll come back to research experiences. So with research experiences for undergraduates, you can apply to be a site that provides research experiences for undergraduates, or you can also apply if you get funded by NSF in a regular proposal. You can apply for funding to help with undergraduate research as a supplement to that, but I'll come back to that. So that leaves three parts of NSF bio that I haven't spoken about, and basically two of those I suspect, well, the first one, this one, is not going to have much traction in this room, but the three are based basically on size. So environmental biology is looking at species level, community level, ecosystems level. I'm just going to skip past that, but if any of your research is at that level, then these are probably the programs that you want to be applying to. Then we get down to organismal level, so aimed at understanding organisms as units of biological organization. And we've got these 5 systems that are then subdivided into clusters. The ones that might be of real interest here are these 3 here. So with neural systems, yes, we fund a lot of neurobiology, not anywhere near as much as NIH, but if you're working on a funky organism, if you're doing comparative neurobiology, if you're working on the neurobiology of sensing your external environment and then transducing that signal, then neural systems is definitely something that you should be thinking about. Developmental systems, it's the same. They're looking at, just like neural, they're looking at evolutionary approaches. They're looking at different organisms, not the mainstream organisms. But that's not to say that you couldn't get something funded out of the mouse or in either developmental or neural. But generally, they're not going to want to fund, like all of us, where NIH has a big footprint. And so where NIH would normally shy away from something because it's not close enough to translational, or it's in an organism that really has got no bearing in human health, so it's not a mouse, maybe not even a Drosophila or a zebrafish, then this is where NSF would pick up the slack. With physiological structural systems, when I was applying to NSF, this is where I get funded from. So they do a lot of things like symbiosis, defense in non, they would do plant defense, for example, or they would do defense in sea urchins, non-regular models that would be common to NIH. And then we have the Molecular and Cellular Biosciences, which is the division to which I belong. And so this is taken from our solicitation, which is currently 15510. And that is about, we've actually written the new version. And it'll probably go up in the website later this year. But it's pretty much the same. We tweaked the language a bit, but we didn't tweak the fundamentals, which is that and this second point really is the major one, we support quantitative, predictive, theory-driven research. And so a lot of the PIs ignore that and just put in stuff that they want to put in. And so this is where writing for the program director becomes important, because if you are doing science, which is, there's kind of two versions of this. There is the one where you've been working on the same protein for 20 years, and basically you're asking for $1 million to buy a bigger hammer to keep hammering away at the same. We will no longer fund that sort of stuff, no matter how eminent you are in the field. We don't fund the study of molecules in isolation. We want you to be able to scale your research, meaning that you might be working on a protein, but you're looking at how it interacts in an organelle or affecting the cell. So there's scalability to your research. So you're not really reducing your whole problem down to a single protein. It's got to be more than that. But we also want quantitative and the second thing that I was going to say, the second type of proposal that we get that we don't like to fund, or we don't often fund, I should say, is where people have a favorite protein, they're using some really elegant genetics perhaps, and they're backing that up with biochemistry, and that's it. What you get is a purely descriptive proposal. Lots of nice pictures, there's no numbers, there's nothing. Where we want to see in molecular and cell biology, especially cell biology part of it moving, is a numbers-driven business where we can start to build models that are based on quantitative biology to make predictions about cells and the moving parts within cells. So we've gone all the way down to the reductionist level, now we're trying to build that back up and we're trying to put things back together again. And so that's why we've given a lot of support to people like Wallace Marshall here at UCSF, not just because that he runs quantitative cell biology workshops, which is great, and teaches a lot of modeling to people. He's also running the synthetic cell biology large program. But it's the whole, he's really embraced the quantitative aspect of the biology. And he's enabling young scientists and mature scientists to build their computational skills as well. And we fund, there's a workshop in Chicago next month for people who have never done any computational biology whatsoever. Bring your big data set and we'll help you work on it. And so we're funding different levels, the community with different levels of expertise to try and embrace computational biology. But having said that, we understand that not everybody feels comfortable doing that sort of stuff. So often the reason many of us get into biology in the first place was we weren't perhaps that great at physics or math. And so if you want to bring in a co-PI whose job it is to do the computational biology, that is fine. You know, that is, we are happy with that. The only time that becomes a little tricky is if you've got a career award. We'll come back to that, but you can still do it. So we're really, especially in my cluster and CDF, we're really big on this element. So very brief, give you a, 1 sentence on each of the 4 clusters. To a certain extent, so what molecular biophysics, and these are the guys who will fund your research if you're looking at from the atomic to the molecule scale. So if you're doing crystallography, if you're doing cryo-EM, if you're doing molecular dynamics, these are your guides. If you're looking at how two molecules interact with each other, that's the scale that they're working at. That's who you should be going to within our cluster for funding. Systems and synthetic biology is the newest cluster, came out of cellular dynamics and function, our cluster. So there's kind of two elements to it. There's the systems level and the synthetic level, as the name might suggest. Systems level, so really looking at regulatory and signaling and metabolic networks. And so the sorts of problems they're looking at is instead of having Afghani farmers grow our opium, why don't we try and make our opiates in yeast? How do we do that? Because yeast can make opiates, but we can maybe move the 10 enzymes that are involved in the pathway into yeast and see if we can get yeast to make our heroin for us. You wouldn't put it quite like that to a senator, but you get what I'm driving at, right? But of course the problem is often when you do that, it fails completely and you don't produce anything. And it's because we don't understand the dynamics of the network. And so it's not just a case of loading a cell up with 10 new enzymes, but you also have to work at the regulation of that network. And so that's what's going on with systems. With synthetic, what we're looking at there is things like DNA with 4 new bases making amino acids that are designed for the process. And so again, looking at how perhaps we can use microorganisms to affect change in manufacturing industry, and using the fundamentals of what we know about DNA, RNA, protein to build polymers, for example, in these artificial synthetic systems. Genetic mechanisms, obvious gene expression, epigenetics, chromosome dynamics. Again, this is potential large overlap with NIH, but it's certainly, they would fund at a much more basic level of research. So they certainly no translational element to that. The one thing I'll say about this is if you're working on the nucleus as an organelle rather than as the keeper of the DNA, so for example, if you're working on the nuclear envelope or you're working on the structure of nuclear pores, anything like that, then you probably want to be coming to cellular dynamics and function because you're treating it as an organelle. And the one thing I'll say about all 4 of our clusters is that what we don't see enough of is research that is driven by evolutionary approaches. So we like the quantitative, we like the model building, but if you can throw in some evolutionary approaches in there as well, we would really love it. That's the sort of thing that really plays well with program directors. Talking about program directors. So in CDF, we have panels on organelle biology, and for example, we fund quite a lot of plant proposals, but often what we fund, and it's usually Arabidopsis, but what we fund is often broadly applicable to animal cells as well. So they might be looking at the movement of vesicles, how do vesicles traffic in cells. So one of the things that you should try and do when you write your proposal is think about the broad applications of what you're doing, irrespective of what you're writing about. If it's only going to affect the one organism that you're working on, and that organism is already a little bit obscure, then that's not going to have much traction. But if you're using that organism for a good biological reason, a good sound scientific reason, and you think that the information it will give you a handle on will then prove to have a major influence across maybe a phyla or the whole of biology, then that's something that you should be communicating to the program directors in your proposal. So we definitely like the evolution. we're getting also more into the origin of cells, just to compact that, and synthetic cells, and that's something I can maybe come back to in the discussion afterwards. And of course, this predictive understanding of behavior. So, really, I put this up for this part. Research with disease-related goals is normally not supported. So, I'll give you a couple of examples of what I mean by that. And really, I think most research, especially if you're working in cell biology, almost all your research can be couched to NSF in a way that avoids the medical relatedness. And I think where people really run into problems is when they get turned down by NIH and they just take their R01 and slot it into an NSF package. We know you've done that because NSF is 15 pages and NIH is 12 pages, so when we get a 12-page proposal, we kind of know what's going on. So, two examples. A couple of years ago, we got a proposal. The title was Studying the Movement of Metastatic Cells in a Tumor, in a Tissue. And they then go on and spend two pages telling us why it's important to study metastatic cells in tumors, in tissues. And we return it without review, because quite clearly, if the motivation that you have is metastatic cells, we're not going to fund that, that's NIH. If, however, the title had been the study of cells moving through a matrix, that would have been fine. And just avoid all mention of tumors and everything else. We have a broader impact section. You could spend 2 or 3 sentences in there saying, this is why it's important. Don't over-egg the pudding, but we would expect to see something in there about the broader importance of your research. Just like people who work in the molecular biology of cell wall deposition in plants will spend 2 or 3 sentences talking about the importance of their research in agriculture. So that's one. The other example I have is a proposal last year where the title was studying improving male fertility through the study of spermatogenesis. And again, the male fertility thing is problematic, and we get 2 pages of description of why it's important to study male fertility. And the problem is if they had just couched it in, we want to study spermatogenesis, that would have been fine. You just don't tell us that you want to study it because your next grant's going to be to NIH. So we don't often return things without review. Maybe for grant, we receive about 1,200 proposals a year. I would say we'd be lucky to rise to 10 proposals that we would return. Most people know this, but they're always very disappointed when we do it. So, related programs that you should also be thinking about. Chemistry of life processes. David Rockcliffe's program. If really the project is being led by the innovations in the chemistry aspect of the proposal, then you should be probably thinking about going here. And we often co-fund with chemistry as well, which is the other thing. If your proposal has got a bit of engineering, a bit of physics, or a bit of chemistry, we can take it to those places and say, help us fund this, and we often do the same for them. So that helps us. Physics of living systems is the other program. Krastan Blagoyev, who you may want to talk to about this. So again, being led by physical principles and physics that underlie biology. So a lot of physicists apply to this program when they're tackling biological questions. Funding opportunities. So the ones that I put up are the ones that I'm going to talk about, but they're basically aimed at single investigators rather than, or with co-PIs, rather than the really big stuff. So the regular research proposals. You've got to check the website. Depending on who you're applying to, the dates are going to change throughout the year. For MCB, Across all 4 clusters, it's November the 15th. Might be a day either side of that. It changes depending on the calendar, whether it's a weekend or not, but it's generally November the 15th. That's our one. You send in a full proposal, happens once a year. iOS have got pre-proposals. I think they take pre-proposals at least once a year, maybe twice, I'm not sure about that, and then they cull that down and they only accept 30% for full proposals. So one thing I can tell you is that we're undergoing review in Bio about how we do this and trying to unify across Bio how we do it. And so one of the interesting experiments that you may have read about in Science or Nature is one of the directorates, it was actually geosciences, did away with deadlines completely and half the number of proposals that they received. So we initially thought, oh, this is just procrastination, it'll double. Next year it didn't, it remained half. And so we've now got 2 or 3 years data that it stays down. A couple of other parts of the agency have done exactly the same and with exactly the same result. We half the number of proposals. And we think there's a group of people out there who basically get told by their chairs, you need to put in a proposal, and so they just, feel out whatever they put in last year, know that there's not going to fund it, put it in. And so we are halving the number of proposals, which means, of course, we're doubling our funding rate. Isn't that good? Bit of math. And so we are probably going to go there in 2019. You'll go, which means you can only apply once a year, but you can apply anytime you like. And as soon as we get enough proposals, we'll form a committee and we'll look at them. So we might do away with funding rates. What that also means is that the pre-proposal system in iOS is under review, and it might also go away for the same reason. So these, the regular proposals, usually they're 3 to 4 years. They can be 5 years, but not often. And the amount that you ask for, which is what people always want to know, is what we always tell people is ask what you need, because unless you're really out of the ballpark and you're asking, so normally we fund anything from, let's say, $500,000 to $1.5 million, depending on the project. If you're asking for over $2 to $3 to $4 to $5 million, that's going to be tough, because if we cut you back, that's really changing the project quite significantly. But if you're not that far out of what we want to give, then we'll just phone you up. We'll fund you, but we'll say we're going to cut you back. What do you need to do to the project to meet this amount of money? And we'll negotiate with you to a certain extent. We won't negotiate the number, but we'll negotiate what you can do with the number. And so ask for what you need, but keep it realistic, right? A lot of people think if they lowball us, that'll somehow give them an advantage. No, it just means if we fund you, you won't have enough money to do the project, and we'll probably know that and not fund you anyway. So really ask for what you need. Substantial preliminary data. I would actually say good preliminary data is more important, and it's not quite the same thing. So research, RUIs, I think there might be people here from PUI institutions, which is primarily undergraduate institutions. And so when we fund these RUIs, they're to institutions which have primarily undergraduates. There's a certain number of people that they can graduate with a master's, but they're primarily undergraduates. Clearly UCSF is not one of these. But if a UCSF, you work with one of these institutions and they apply, then you can be funded through an RUI as well. And what that gets you, and I know one of the problems that you have at a medical school like this is access to undergraduates is quite often the problem. But getting a tie-up, with San Francisco State, for example, is one way to give you access to both undergraduates and, as important, minority undergraduates. And so RUIs, again, is something, if people want me to speak about it, I'll come back and do that. But it's just generally a regular proposal. We don't expect you to do as much as if you were applying from UCSF because you're going to have a higher teaching commitment. And so the panels know to expect maybe just a little bit of less of a burden on the PI works. But again, it's something to get back to. The eagers, which people, as soon as they get their decline from us, I always get a lot of people phoning up and say, well, I want to put in an eager. But it's not quite that simple. You have to be allowed to, we have to give you permission to put in an eager. And basically what the eager will give you 300,000 for 2 years, so people see it as a way of keeping the lights on in their lab. But not only does it have to be transformative research, it has to be research which would not survive well in the panel system, meaning it will not review well in the panel. And that usually means it's because it's cross-disciplinary and the panel won't get it. Not that the panel didn't get it because they didn't understand your science in the first place in the last panel review. And there's a few other caveats to how we produce the eager as well. But definitely it's going to be high risk, high payoff as well. The other thing is we don't expect preliminary data, other than maybe, you can maybe put in 1 or 2 bits of preliminary data, but it has to be exploratory work in its early stages. So those are all things that that go towards an eager, successful eager. Rapids is where we have to, as an agency, respond quickly to a crisis, either man-made or natural. And a good example of that was NSF, was the first agency to respond to the Zika crisis, and we provided grants to modelers who then modelled the disease to tell us where the disease was moving to and where it come from and everything else. So that's rapid response research. Careers. So if you have just got your tenure track position nailed down, you're probably thinking, I've got 6 years, which is great. I'm going to apply for a career. A couple of things you know about careers. You get 3 shots. If you're on a 6-year tenure clock, you do not want to be doing this in the first 2 years. You don't want to be burning up shots at your career award that would be better done once you've matured your project. You know what questions you want to ask. You've got a ton of preliminary data. You're in a good spot. 3rd year in, this is when you should be thinking about career. If you get your career award the day before you get tenure, we'll give you the career award for 5 years. If you get it the day after, you don't get it. So that's how it works. Minimum funding for 100,000 per year. Nobody ever asked for that for 5 years. Typically, careers would be closer to 1.5 million, and always 5 years. Substantial preliminary data. Got to integrate research with the educational component. I'll just come to that in a minute. And what I would suggest if you're in a position about thinking about a career or you've just got your tenure track position is apply to the November panel first. The panels are structured so that once you get your decline from a November panel, if it is a decline, that you've got enough time to turn it around into a career. And so you get 3 sets of eyes on your proposal, you work out where the big problems are, and you turn that into your career. Or you spend the first 2 years just going to the November panel and getting different sets of eyes on the proposal. And that way you can bounce between the panels, that's not a problem. And most careers, some get funded first time out, sometimes it takes a little bit longer because they're ironing out those problems. You can have a co-PI, but you can have collaborators. So if you want to bring in somebody for the modelling, you can do the whole collaborator bit. The race, for those of you who remember Inspires, this is a replacement for the Inspire program. High risk, high payoff. It's going to cross-cut across different areas of science. We don't review, normally it has to get to be eligible, it has to be accepted by 2 program officers from different parts of the agency. So bio and physics, for example, one I'm involved in. And again, it goes up to $1 million. So it's not a huge amount of money over 5 years. But this is where a lot of the interdisciplinary research goes. If you want to know more, funding opportunities in the NSF page, top here. And I would ask you, look at the most recent solicitation and read the solicitation. It's associated with every funding opportunity to make sure that you're hitting the mark. But also, and I really would advise you to do this, get a 1-page summary together of your proposal and send it to the program officer of the program that you think is the most appropriate. And if he or she is doing their job properly, they will either say, yes, this is appropriate, or they'll shop it around until they find somebody that is appropriate. So they should be there to help. Now, if you phone the program director and MCB, they'll get back to you quickly. Our proposal load is actually not that heavy compared to our friends in, for example, engineering, who have 5-fold heavier proposal load than us. They barely lift the head from their desk to answer email or answer the phone. So I would, if it's biological, I would definitely go bio first and then let us shop it out. And if you want to see what we're funding, you can do that as well. You can look at awards as well. So there's a NSF award database that you can use. So the last thing I want to touch on is the proposal itself. And this is a bit that kind of spooks a lot of people, especially if they're used to writing R01 proposals, because we have this broader impact. And people don't know, if they do know about broader impacts, they often don't know how much they have to emphasize that within the proposal. You've got 15 pages. How much, one of the questions I get most often is, how much do I have to write on broader impacts? So the answer is what it takes, but usually it's about 3/4 to 1.5 pages. So we have these 2 criteria. To be funded by NSF, you have to be excellent in both criteria. And so I see a lot of proposals fail because they have got really great science, but they ignore the broader impact. In which case, we cannot fund you. In fact, you go immediately to low priority, and you cannot be funded. So you have to be good in both the intellectual merit and the broader impact. The intellectual merit is the science, really the guts of the proposal. The creativity, the originality is what's going to help there. Advanced knowledge across fields is going to be important. We also look at the qualifications of the investigators. Less important if you're a new investigator, and your access to resources, and clearly at UCSF that is not going to be a problem. And usually it's not, but occasionally it is. The broader impacts. This is where you want to be taught that it's the educational component of the proposal. And so we have this saying at NSF, the Noah's Ark approach to broader impact. And a lot of PIs take the Noah's Ark approach, and it's not a problem, okay? Sometimes it's a problem with the panels, but we try and sort them out. So Noah's Ark is 2 high school students, 2 undergraduates, 2 graduate students, okay? And then you talk about the educational component of that. And you know, that is actually fine. So we always tell the panels, you know, it's not an arms race. We don't need bigger and better, broader impacts every year. As long as they're having an impact, that's what's important. And so we try and tell them that. And so often you're going to be looking at bringing in minorities. Now, often it's undergraduates, and so this is where you might want to hook up with one of the colleges or universities in Northern California, or California, wherever, but even have to be in the state. But it can be high school students, and it can be high school teachers as well. And there's different ways, whatever way you choose to go, make sure that you've got a letter saying, you know, if this grant gets funded, then I will certainly be willing to help, you know, Dr. So-and-so with his broader impact from the head of the school, the high school or whatever. Some people do middle school, some people even do primary school. There's all sorts of ways you can do broader impact. If you've got kids in primary school, there's a population right there that you can connect with. And so, you know, that's fine as well. You just need some letters of support saying that they'll support you if you decide to do it. You just can't pull it out here and expect them to do it. With undergraduate education, there's often within universities already set up groups that will help you with that and help you with boilerplate to do that. Sometimes, and this is especially true of careers, you have to do outreach to the community. And so that could be setting up, as one of the proposals that I funded, career proposals, setting up an exhibition on microscopy at the football stadium, at the college football stadium, or setting it up in a museum, or setting it up in anywhere where the public meet in large numbers. It could be at local Safeways, as long as you're taking science out to the public. But you don't need to do all of that. You only need to do elements of it and what makes you feel comfortable. The important thing is we're asking you to do broader impact. We're asking you to think, how do I know if I had an impact? And so you have to be able to quantify your impacts. And so that's another important thing that you should be thinking about is how will I know I've had an impact? Some people use quizzes. You've got undergraduates coming in, ask them what they want to do, what their career expectations are. They work in your lab for a year, ask them the same thing when they leave. Has it changed? How many of them went to graduate school? That sort of thing you can quantify. So these are just some examples, and basically I spoke to that, writing articles, and I was running a blog, science blog. That would be a broader impact. So, you know, there are different ways to get to this. Mentoring, seminars, workshops. I've actually spoken mostly about that. So these are just the last few slides. Writing a successful proposal. Speak to a program director, speak to a program director, speak to a program director. I cannot tell you often enough. Read the solicitation. Understand who you will read your proposal. Yeah, it's going to be 2 or 3 panelists who may not have a background in your biology. So make sure that you point out the obvious to them. Don't assume that they're going to know everything about your science. They won't, I can tell you that. A lot of PIs are very disappointed to phone me up and say, why did they write that? Everybody knows that. Well, clearly everybody didn't know that. All right. So think about that. Don't be afraid just to point out the obvious. So understand who will read it, but also think about the program directors as well. So you'll also have to write a data management plan, postdoctoral mentoring plan. You'll probably get boilerplate from your institution. I don't mind you using it, but try and personalize it, especially for the postdoctoral mentoring plan. If you're asking for a postdoc, try and make it about, even if you haven't got somebody in mind, try and make it about a person and not some institutional boilerplate. It just reads a lot better. You will not be, unless you miss them out completely, we won't not fund you because of these. We'll ask you to rewrite them if we're going to fund you. But, you know, it's the writing defensively. Once a panel finds something to criticize you about, they get on a roll. And so this is 2 areas where they can really hit you hard. But it's not too difficult to write good versions of these. So got there. Yeah, big picture, broad significance is always good to frame whichever project what you're working on with big picture. you know, NIH, they've got the nice first page that you have to do, the specific aims page. So if you can take a couple of pages to effectively get everything on that, what's on the first page of an NIH, into the first couple of pages of an NSF, that is often a style that works really well. We want to see hypotheses. A lot of proposals come in, never state their hypotheses, although they're clearly working to one. That is a mistake. You should also, you know, we also see people with hypotheses, but the experiments that they suggest are not going to test the hypotheses. That's a big problem that we see over and over again. And distinguishing amongst competing hypotheses. So we often have, their panels often pick up on, well, yeah, I'll tell you about one thing, but it's not going to tell you about another. So the hypothesis part is very important. Another thing that people miss out, and it's absolutely mind-boggling to me, that people miss out telling us what organisms they're working on, or cell line. They just assume that we know, especially if they're working on cell lines. They just never tell us that they're working on a cell line. And so, you know, if you're working on some obscure lizard, then they're only too quick to tell us what they're working on because they know that we like that. So that's another thing. You know, that should be able to get that out right away, what you're actually working on. So this is the last slide, I think. The reason proposals fail to get funding, absence of an innovative idea. So when we sit in panel, there is a board on the wall that says, if the research is incremental, it automatically goes to low priority. And as soon as we hear that incremental word being in a discussion by the panel, it dooms the proposal. And so, you know, it's like asking for the bigger hammer to work on the protein that you've been working on for 20 years. We don't want to see incremental research. We want to see you taking big leaps in your research, especially big risky leaps in your research. So it's got to be exciting, it's got to be cutting edge, I mean, that should go without saying. Mistakes people make: unclear expression of aims, faulty logic, less than rigorous presentation. Obviously, interdependent aims. You can't do aim 2 unless aim 1 works. That's an automatic fail. So your aims have to be independent of each other. Badly edited proposals really do not do well. They never do well. No matter how good the science is, who is submitting it, they never do well. This usually not a problem, this part of it. Uncertainty concerning future direction. If it's not obvious where the next stage of the research will go, then we will have problems funding you. We don't necessarily fund open-ended research, but we like to see your research does have some direction. Another reason that you don't get, another common error is that you forget to put the broader impact section in. You send us the wrong version of the proposal when you, or at least you upload the wrong version of the proposal. There's a page and a half missing from the end and there's no broader impacts. Then we'll review it, but it's never ever going to get funded. So hopefully that gives you some idea about NSF, some idea about what we fund. These events are great, but it's much better to speak one-on-one with people about their individual problems, and your program director should be more than willing to pick up the phone or answer an email to do that, because often the problems are very specific problems. So again, I would encourage you to contact your program director. And with that, I'll happily take questions.
So you mentioned it's very important to talk to the program director, but then you also mentioned that you have half of you are like in a rotating position. Mm-hmm. Does it matter if you go to the permanent one or the rotating one, and how do you distinguish?
So if you're unlucky enough to pick up somebody their first day or the first week or the first month, it can be really scary for them, because they're not used to dispensing advice and wisdom. But usually once people have been in the job 3 or 4 months, and especially if they've gone through a grad cycle, then they know pretty much most of what there is to know. But they will always defer to a colleague. If they don't know, they should say, I don't know, and go and find out from a colleague. So the rotators, probably the greatest fear of rotators when they come in is picking up the phone, just for that reason, and phoning PI to tell them what they're trying. But it shouldn't be a problem. It shouldn't be a problem.
Is there any sort of implicit bias against work being done in human tissue or human cells if it's not obviously biomedical in nature?
No, I have to say that I actually did an analysis of what we fund, analysis of what we received from what we fund, and I would say about 30% of what we received was on mammalian cell lines. I didn't break it down into human. I would say not a lot of that got funded, but it's not because of implicit bias against it. It's because there is an ultimate that we worry about. The science that was being submitted was often stuff that had failed to get funded at NIH, and it was clearly NIH type of work, and so we try and avoid that footprint. But if you're asking the right questions and framing it in the right way, there is no reason whatsoever that we will not fund that sort of research. But I understand where you're coming from.
So is there any award like specifically for junior PI who just get started their own labs?
There's a career award, but that's like
Later stage.
standard award. You just compete in the standard pool. But I will say this, we do look to fund new PIs out of the standard pool. So when we're making our funding decisions, we're looking at geography, we're looking at minorities, we are looking at how new the PI is. But that doesn't mean to say that if you're an old white guy from UCSF, you're never going to get funded. That's not the case. We fund a share of senior PIs from senior institutions as well. But we do cut you a break when we're looking at what you should be funding.
I just wanted to ask you, to what extent does NSF have any collaborative efforts with other funders? Not necessarily NIH, but any other.
Actually, not as much as I thought going in. We have the odd program with NIH. There was one in ecology and in Unity, I vaguely remember. We had a program with USDA, which was for eager awards, but really not that much, and not as much as I thought it would be.
And sort of as a follow-up to that, is there something that's kind of an equivalent of a common fund? So something that's sort of a pool of discretionary funding where, you got something kind of creative and outside of your usual box, although your usual box is pretty open, but where there might be ways of bringing funds together for that?
That's the sort of thing that would probably be above my pay grade, unless there was an obvious awarding mechanism I could point you to. You probably want to speak to the Deputy Division Director, which is my boss, about that sort of stuff. But everything has to be associated with a proposal, and the proposal is usually associated with a solicitation. Yeah. So I don't think there is, but...
I have a question on the funding increments and kind of the consideration where I've seen an NSF award where the funding is released for all 3 years all at once,
Yep.
beginning, and another one where it's incremental yearly.
Yeah. Why do we make those
Yeah.
decisions? So it's all about, so this is one of the things that Cluster Leader does is he takes care of the spreadsheet right through the year. And so at the beginning of the year, I get a budget of 35 million or something like that. It's up to me to make sure it all gets spent out. And so what we do is when we fund most of our stuff, at this time of year. We fund most of it as a single tranche of money to the investigator. And that means the money gets spent. But to increase the amount of money we spend, some grants we do as these CGIs, incremental, which means we release it year by year, which means that instead of funding you $1 million this year for a 5-year proposal, we do $200,000 a year. So I'm now spending some of 2018, 19, 20, 21's money, right? That's called our mortgage. We don't like our mortgage to get too high because that means at some point we'll run out of money. It's like a Ponzi scheme. And so if you're a new investigator where there's an element of risk, or if it's a career award, we will fund it as a CGI just so that we can claw back the money if you don't get tenure. Okay? Because a career can't be transferred. That's the best example. So it gives us the ability to claw back the money. If you're a regular PI and we're not having any financial concerns about you or your institute, which is another question entirely, then you'll almost certainly get the money up front to spend it as you wish. That's the difference.
So you mentioned that it's really important for NSF to stay away from the footprint of NIH, and as
Or part of NSF.
okay, so and as people who I help people who normally write grants to NIH. So if I'm advising somebody to write an NSF grant, could it be helpful to include language in the proposal that describes how this particular proposal is outside of NIH's footprint and therefore more suitable for NSF?
You could do that, but I'm not sure that I would want to burn too much space doing that. We normally read them and we reach a decision pretty quickly if this is not for us. And it's usually fairly obvious. And it's a high threshold. We don't return a lot, as I said.
For these eager awards, one is not expected to have a lot of preliminary data, but none.
It could be none.
So
be a
could it
little.
be just this great, fantastic idea that theoretically should work?
Yeah.
And so you don't have to present
Yeah.
preliminary data?
Yeah.
Okay. And then what would be the process for that?
You get in touch with your program officer and say, I've got this idea, submit a 1-page, and they'll look at it and say yes or no. If they're more likely to say no than yes, because this is the threshold. Eagers do not go for outside review. So they're not beloved by our committee of visitors because there is no community involvement in their review. It's the program officer who reviews it needs to get one other program officer to sign off on it, and that's usually a formality. And so it's the acceptance of that 1 page is usually the hurdle, and we tend not to fund too many. They really have to meet our criteria. Yeah.
So I was very interested in your advice for the career to maybe have the PIs apply first to the regular November cycle to get some reviewer feedback already. But we are also a research development professional. We spend quite a lot of time explaining to our PIs that the career award is quite a different beast than a regular award. And so basically you're saying maybe it's not that different?
I don't think it is. So it's 5 years work, right? And so you're maybe trying to fit an extra aim, for example, in compress it. And we expect a bit more from the broader impacts. So we expect broader impacts to be integrated, the research and the education to be totally integrated. We expect you to be doing a bit more. There's usually a bit of community outreach there. There may be working with high schools, and there's maybe some undergraduate involvement and graduate student involvement. Yeah, so there's maybe a bit more involved. But I just reviewed a regular proposal from somebody who had a career turned down. I reviewed it, and it was basically the career award resubmitted, like I was saying. And I decided this time he'd done enough. Let's fund it. And what I did was I went back and resurrected his career award and funded that because it's more money. And funded that But essentially, it's the same proposal. So no, it's not really a significant advance on 15 pages. So we're looking for the same stuff. It's just going to be, there's a bit more data, probably a bit more work in there.
Can you say something about post-award progress reporting?
Yeah, if you have a CGI, you want to get your progress report in time because we won't release the CGI, we won't release your funds until we sign off on your progress report. Most progress reports address what you've been doing with the science, definitely address what you've been doing with the broader impact, upload any papers that you've published as a PDF, and really should not be a problem after that. But and if you're applying for funding and you've got an outstanding report, or your co-PI has got an outstanding report, or your co-PI has a co-PI on another grant who has got an outstanding report, that's all going to have to be reported before we're going to release money. So that gets flagged automatically before we can give you money. That any outstanding, even in the sort of family tree of your proposal, any outstanding reports have to be submitted. So normally that's usually the final report.
I have one more question about the idea of getting rid of the deadlines.
Yeah.
So you mentioned briefly that then once you have enough proposals coming in,
Yeah.
you'll put up a panel. So how do you advise PIs on their timing? What's
So
time frame
we
when
don't.
they
We
expect
well,
to
we won't.
start it?
We won't do that. So normally we have 5 panels in March, one for different areas of CDF. What we'll now do, I think, because we're just beginning to think about this, is irrespective of where the proposal comes from, it will all be held in one panel, and we'll just get the expertise that we need in that panel, and we might make more use of ad hoc reviewers as well to get specific expertise. So yeah, the way it's going to work has not been worked out, mainly because we would prefer, I think, to stay with standard dates. Everybody knows where they stand at that point. So we're trying not to think too hard about it at the moment, but it's got to come. The other thing I should mention is, if we don't think there's anybody on the panel at the moment, if there's nobody on the panel that we think is experienced enough to review your proposal, we'll send it out for ad hoc review, as well as the panel. There's a bit when you submit your proposals that asks you to provide the names of reviewers. Please make use of that, because that's the first place we go when we're looking for names of people to review your proposal if you want ad hoc. And some proposals are difficult to get ad hoc. So it's great if you can supply names. They shouldn't have published with you in the last 4 years. They shouldn't be your best buddy in the usual conflict of interest rules. But if you can suggest names in the field, that's great.
Towards the beginning, you said the award rate was like 12 or 15% on the regular
15%.
proposals,
Yeah.
but that invited proposals had a much higher success rate. What are examples of this invited?
The eagers would be
Eagers.
eagers, rapids, and eagers, rapids, raise used to be the
Okay.
inspire. Anything where you need the program director's permission to submit the proposal is usually got higher funding rate.
Thank you again to Dr. Cunningham. These insights are critical as we work to maximize the funding that we get from NSF. So I would like to invite everybody to join us just outside these doors for a reception where Dr. Cunningham will be present if you'd like to speak to each other or to him more. Thank you very much.
Thanks.
Yes, yeah. It was
Oh, the GSI thing you mean? Integrative Organismal. Yeah, but it's like got field ways. Yeah. I mean, I don't know, it sounds like they're talking about setting up thing in some modified form, but like, I feel like the folks at the top I'm so loud. I don't know, I'm like part of that small community I'm glad that's well, there's nothing logical about but I think I think that was the argument about like I think that that's exactly
Yeah. So everybody knows
You did say there are
well,
so the funding coming
I'm done. And that all sounds So remember we went for our half-day workshop. So put them over like I said,
Would you be interested?
So I've taken on.
You wanted to come up here and then make it more. Right. Well, actually. Oh, just for the. But I'm listening. I don't have
That's us.
Do we want to share our name?
Yeah, I think I remember that.
Don't send me anywhere, so I'm starting to I wanted to see if
So I'll say right? Yeah. Talk about it.
So maybe see how we make it.
I want us to have another one because I know they love
At that time, we used to
talk about that.
Okay, thanks for telling me.
I was so happy to hear you guys were here. I was just